Today, I'm going to actually present some of our recent work. This is the first time I've actually presented this on a new problem and application to succinct data structures. And in particular, it's about designing baits for DNA enrichment. So antimicrobial resistance is still receiving a lot of media attention, and predominantly because experts are concerned that the pandemic could undo much of the progress on antimicrobial resistance, or AMR, especially in hospitals. In fact, the relationship between COVID and AMR is really complex. Currently, over 10,000 people die worldwide from resistant infections, and this has been recently rising. So the traditional method to identify resistant bacteria from these samples is a culture-based one, which involves isolating the bacteria on general or selected media, and then assessing its growth in response to specific concentrations of antibiotics. Unfortunately, these culture-based methods can only identify the presence or absence of resistant bacteria. On the other hand, metagenomic sequencing, which aims to sequence all the DNA within a sample, can be used to more thoroughly identify all the genes and species in microbial sample, or microbiome. Thus, metagenomics can also be used to identify all genes and mechanisms that makes a sample resistant. We refer to this as the resistome. So currently, it's believed that there exists around 8,000 known genes or mechanisms that make bacteria resistant. However, the existence of the community of mechanisms and genes that make the bacteria resistant is very complex. On the left and on the right are graphs illustrating the number of AMR genes in a set of metagenomic samples. So for example, on the left, we see a heat map, and we see 5 different types of samples. So feces, soil, water, truck, and plant. So this is from a food production facility. So by plant, we need per production plant. And here it's a heat map. So we're seeing the specific genes and their phylogenetic relationship to each other. On the right, we have a collection of metagenomic samples, but here we have them separated out by mechanism and class of resistance. So what you're going to notice here by these 2 graphs is that there's regions of that are of high abundance to AMR, so specific samples, but also specific types of resistance. But then you're also going to see extremely low abundant regions. So if you look at plant, for example, we actually were unable to detect a single AMR gene or mechanism within those collection of samples. On the left, where you see it separated by class or mechanism, you're going to see that there's some classes or mechanisms where we're seeing extremely low abundance. So on the exit, for example, we see that we were unable to detect any AMR genes from a specific class. Now, it is unclear. What you're going to notice, I said, is that we were able to identify. It is unclear if the genes were actually there or not, because metagenomics is not a perfect science. So shotgun metagenomics is actually rarely used for public health surveillance. And one of the first reasons is that that was pointed out in the last slide, which it suffers from very low sensitivity. So we found that only on average between 2 and 5% of the metagenomic reads actually align to the AMR database. And this value can get as low as 0.1% if you look at things such as water samples. So what this means is that you have rare or low abundant reads, you're not going to be able to detect them, or it's going to be very difficult to detect them using metagenomics. This second sort of issue with metagenomics is that it does suffer from a lack of context. So shotgun metagenomics is often paired with short read sequencing, which requires the DNA to be highly fragmented. And the context of where these genes occur, i.e. in which chromosomal DNA or which plasmid, has a public health outcome. So trying to reconstruct where these genes occur is very challenging from shotgun metagenomics. So we're working on both of these challenges, but today I'm actually going to talk about some of the results we have concerning ameliorating this low sensitivity. So there exists a number of different methods that can enrich for targeted DNA in metagenomic samples. One of the methods I'm going to discuss today is enrichment via DNA probes. DNA probes, or we also say DNA baits. So given a database of genes or microbial species that a biologist is interested in, it is possible to slice up the database into k-mers, that is every k-length subsequence in the database, and create synthetic probes from these k-mers. Then these probes are then applied to a particular metagenomic sample, and then they bind to the targeted DNA. Any unbound DNA is then rinsed away. So what supposedly remains is just the targeted DNA, the bound DNA. And that DNA is then left for sequencing. So we have applied this technique to enrich for AMR genes, and it worked successfully. Here are the results that we had from a prior study. And here we used short reads. What you're going to notice here is that we are able to enrich for a lot more AMR genes that were seen in the unenriched data. So in the unenriched data, we saw 247 AMR genes. And then in the enriched data, we were able to identify 1,441. And in particular, we were able to identify rare genes that we hadn't seen before in standard metagenomic data. But here, what I'm going to mention is that the process of creating the k-mers from the database is purely a computational one. Now, if you have a small database, such as our AMR database that we are looking for, then you can take that database and apply a k-mer counter, find all unique k-mers, and then use that as a template for your probes. Unfortunately, the goal becomes a lot more challenging if we, once we move to larger databases. So here we have to take into account that the probes bind to everything that has at least 70% sequence homology. So we don't have a one-to-one relationship. The second thing is that we need the smallest possible set. So attempting on optimizing for both of these things is quite challenging. And just to highlight the second thing, I'm going to mention that biologists purchase these probes by tier. So on the first tier, you have approximately 57,000 probes, and then it goes up from there. On the last tier, you have a maximum of 230 probes. So you really want to reduce the total set of probes, because that's going to have a direct impact on the cost of the experiment. So that's the first thing. The second thing to point out is that you do have a maximum of 230 probes. So there's not any way that I'm aware of that you can somehow gain more probes per experiment. Now, as I mentioned, this becomes more challenging as the database increases. So keep that in mind when we discuss the next slide. So from the biologist perspective, the goal is to design a set of probes or a set of baits with no more than 230 total baits that together target all the 120 mers within the set of reference genomes, and to accomplish this in a reasonable time frame and a reasonable amount of memory. Now, the genomes that the biologists were actually interested in, or my biology collaborators were interested in, are the following genomes. So we have around 76,000 Salmonella genomes, approximately 60,000 Campylobacter genomes, and so forth. So we have these sets of thousands of bacteria genomes. And what they actually want is they want a bait set that is going to enrich for all of these genomes. So they don't want 4 different bait sets, but they want one bait set that is going to enrich for all these genomes. So the first thing that we wanted to do was to survey the current work and see if there's an existing method that can actually accomplish this. It turns out that we're not the first people that have studied this problem. So there's an existing approach in the literature. It is referred to as CATCH. So what CATCH was developed for is to enrich for human viral genomes. So they have a collection, quite a large collection of viral genomes. And then they do probe design to enrich for all of these viral genomes. So this was published in 2019 in Nature Biotech by another research group. So the first thing that we attempted on doing was saying, well, how about we try and use CATCH? What is going to be the outcome? So we tried small sets of our bacteria genomes, 2 types of our bacteria genomes. So 30 of one type of bacteria and around 200 of the Salmonella genomes. And what we saw from the scalability was that time was the bottleneck. So in theory, it would take over 4 CPU years for just a small for the Salmonella, for the probes to be designed for the Salmonella genomes. So we decided that, well, it inspired us to do 2 things. Number one, it was interesting to ask, well, what is the computational complexity of this problem? In some ways, it seems significantly harder than some of the problems that I've previously looked at in bioinformatics, harder from a computational perspective. And then to ask, well, can we improve on the existing methods? So the first thing that we attempted on doing is coming up with a combinatorial formulation of the problem. So our formulation depends on the following definition. So we have a set T of sequences. So these sequences are going to represent the baits. And we say that T is going to theta, or theta covers a set S. A set S is going to be another set of strings, where S is going to represent our database of genomes. And we say that T theta covers S if for every subsequence of length L within our database, there exists at least one sequence in T that has Hamming distance at most theta from it. Now, what we illustrated on the left-hand side here is that the idea that more that maybe, which is maybe apparent by now, which is more than one bait can cover more than one sequence and more than one subsequence within a sequence. So this is where the idea of trying to minimize the number of baits comes into play. So our formulation, which we refer to as the minimum bait cover problem, takes in 3 inputs. So the first is 2 integers, theta and L. And the last one is just our set of strings, so our database of genomes. And we assume that the database of genomes is over a finite alphabet, sigma. Then the question is, what is the smallest possible set T of L length strings such that S is theta covered by T? So this is our formulation. So given this particular formulation, we were wondering what the general computational complexity of the problem is, which led us to the following proposition. So the proposition is as follows. We showed that the minimum bait cover problem is NP hard, even for n is equal to 1. So that is, we have a single input string. Theta is equal to 1. That is Hamming distance equal to 1. And L is equal to 2. So that is your baits have a length 2. So given this hardness result, this pretty much rules out any fixed parameter tractable or slice-wise polynomial algorithm with respect to n, theta, and L. However, we should point out that this result requires, in the proof, it requires that sigma is somewhat large. So since we are primarily interested in instances of the problem where sigma is equal to 4, this might leave some hope for small alphabets, for there to exist an efficient parameterized algorithm for small alphabets. So we were wondering if we could strengthen this result, and we accomplished this. We came up with the following theorem, which is as follows. So for every value of k greater than or equal to 2, the problem is NP hard even for sigma equal to k, S equaling to a single input sequence, theta equaling 0, and L, that is the bait length, equaling to log the length of S. So what this theorem implies is that the problem remains intractable even for extremely small inputs, i.e. the DNA alphabet, a single input string, Hamming distance of 0, and the bait length being log S. Indirectly, this also solved a existing open problem in the literature that is not related to bait design. So this is our theorem. So given this theorem, we realize that we have no hope of solving this problem exactly in polynomial time, even for extremely small inputs. So the only hope in solving this problem and helping my biology collaborators is to come up with a heuristic solution. So we tried several different or discussed several possibilities. We even partially implemented some of these possibilities. The first one that we took a stab at was to build the De Bruijn graph, find all unit takes, and then attempt on trying to find some set of L-length subsequences on the unitics. So essentially, you're using the unitics to break down the problem to a smaller input. Another possibility that we are looking at is dynamic programming algorithm. We know we don't have any guarantee, but if we could use the output of the dynamic programming to somehow bootstrap another heuristic solution. We also considered expectation maximization algorithms. There were a number of different ideas that we tried. Not saying that none of these possibilities could work, but we tried a slightly different direction. We asked ourselves, what is the simplest possible solution? So the simplest possible solution is just to slide a window of length L across every single one of the input strings and ask ourselves, if the starting position is not yet covered, then we're going to take it to be within our bait set. If not, we're going to go on to the next one. So this is the problem this is the direction that we were going to take. And what it essentially does is it then boils down to a data structures problem. Why is it a data structures problem? Because we need to efficiently decide which other sequences are going to be covered by that new bait. So our solution, I'm going to briefly go over. I hope I have enough time. I know we're going a little bit over. So the first step is to construct the FM index and the generalized suffix array on the input strings S1 through Sn. And then we're also going to store a bit vector for each position of each one of our input strings. That bit is going to then determine, is it covered or is it not covered from our input? So we consider each position of every string, as I said, and we ask ourselves, is that position covered by a bait? So we need to simply check our bit vector in order to answer that question. If it's not, then we're going to consider that particular position and take that bait, take that Elmer starting at that position to be contained within our bait set. So we're not finished yet because we now need to use the FM index and the suffix array to then find all new positions that are going to be covered by that bait. Normally, this would be easy, except for the fact that we have this theta Hamming distance. So we have to find everything that is within theta distance from our new bait. So how we do this is that we essentially do a seed and extend approach. So we use our FM index to consider all k-mers contained within that new bait. And we search for it using our FM index. Once we find the locations or the range within the FM index, we're going to use the suffix array to look up the particular positions and attempt on extending that k-mer and determine if the subsequent sequence is within Hamming distance theta from that sequence, from that bait. If it is, then we're going to set the associate bait. If not, we move on to the next one. So this is our process, our heuristic. We implemented this algorithm. We refer to it as SWATI because, and I'm probably pronouncing this incorrectly, because it's Finnish for the word bait. We ran our method against 3 different increasingly large databases. So the first one is MegaRest, which is an AMR database. Myself and my biology collaborators actually maintain MegaRest. And then we have our bacterial genomes, which I already mentioned. and then our viral genomes from the Catch paper. We ran 3 methods, so SWATI, Catch, and another competing method, Mr. Bait, which was done prior to Catch. All the methods were able to successfully run on Megarest. As I mentioned, there's about 8,000 AMR genes within Megarest, so it's small enough all the competing methods were able to run. The bacterial, so SWATI and Mr. Bait were able to successfully run on the bacterial genomes. Catch was unable to scale on the viral genomes. Catch was also unable to scale. So as you can see, the running time was the largest constraint for Catch. So here we're going to look at the results more closely for the viral genome. So this is the set of viral genomes. What we're going to, and we're showing the running time, memory, and the number of baits for increasingly larger subsets of the viral genome data set. Here, our input length is going up by base pair. So Catch was only able to successfully run on approximately 8% of the data. After that, the running time became too imperative. Mr. Bait was able to successfully run on all the input. However, the challenge with Mr. Bait was that it does something along the lines of K-mer counting. And because of that, what ends up happening is that it ends up with an enormous number of baits. So I think we have like around 9 million baits for the viral genomes. So remember, our max constraint was about 230. So with 9 million, there's like no possibility to even break up that bait set and do a smaller bait set and do something with it. So this was the constraint with Mr. Bait, that it ended up creating far more baits than what is practical. The other consideration that needs to be made, I'm not able to go to my, okay, there, is looking at the coverage produced by the baits. So what we did here was we took our bait set and we aligned all the baits to the references, and we looked at the coverage. Here we looked at Megarest because, as you remember, the methods were able to successfully run on all the methods on the complete data set. As you can see, so as I mentioned, Mr. Bait produces a significantly large number of baits. So here I want to mention that you want the value to be as close and frequently at 1 as possible. So anything greater than 1 is deemed as kind of inefficiency within the bait design. So here, as we can see, we had to graph Mr. Bait on a separate graph here because if you'll notice on the y-axis that it goes all the way up to 350. And this is again because it's doing something along the lines of counting. If we look at the comparison with Catch and SWATI, they're a lot closer. Still, Catch is producing a lot more inefficiency within the bait design, but it does cover, as far as I know, it does cover all positions within the database. Similarly with SWATI. So I think the coverage was on the order of 99%. So future work. So this is the first sort of attempt at looking at this problem. we're currently using SWATI, the SWATI design base on the bacteria to actually process USDA samples. So as I mentioned, my biological collaborators are actually interested in enriching metagenomic data for these genomes because they're trying to use it in a commercial food production facility to monitor foodborne outbreaks. We've actually paired the bait enrichment with long reads. And that work is currently in submission to microbiome. This is kind of getting back to the secondary problem of trying to co-localize the AMR genes with their plasmids or chromosomal counterparts. And we've submitted SWATI to ISMB, and the paper is also on bioRxiv. I think that there's a number of different open problems here. As I mentioned, there's many different approaches that could work, probably. And I'm really excited about this because I think it's like 50% computer science and 50% biology. And one question that's kind of left open from a computer science perspective is that if there exists any approximation algorithms. So there does exist an intricate relationship between parameterized complexity and approximation. So it's possible that some of the results in this paper might be used to determine the possibility of an approximation algorithm. So this work was a team effort. The first author of it is Jarno Alenko, Ilya Silzori, who is Noel Noyes' PhD student, Daniel Lokshanov at the University of California, Santa Cruz, Travis Gaggi at Dalhousie University, and Noel Noyes at the University of Minnesota.